Okay, thank you very much. We've heard a lot about the Institute today, so I won't belabor it other than to say that at the Institute we've taken a systems biology approach to the translational research program. And what I mean by that is shown schematically on the next slide. So what we do is we consider that since CFS is a systemic disease involving many systems in the body, the brain, the immune system, infections, the gut we've heard about today, we want to look at as many systems as we can at the same time. And so what we do is we involve clinicians, in this case Dan Peterson is our first cohort, and we isolate samples. We isolate DNA, RNA, plasma, and whole peripheral blood cells in a very standardized technique. And we use those samples in a collaborative effort, the exact same sample from the exact same patient at two different time points, about 6 months to a year apart. And we sent those samples for viral technologies, which I'll describe in the first vignette, and that viral microarray will tell you the entire mammalian infectious virus species in that sample. In the plasma sample, we're looking at viral immune pathology by immune profiling a multiplex proteomic array where we look at 30 cytokines, chemokines, and growth factors together. And in the third study I'll talk about, we're talking about genotyping. So who are individuals who are predisposed or on a genetically susceptible host, these viruses and the immune system can't control them. Garth Nicholson talked a lot about the microbial pathogens. Others have talked about individual viruses, but we haven't talked about the immune system as it relates to the pathogens. So we all have a lot of pathogens in our body, and we exist in a homeostasis with those pathogens in such a way that we have a balance. But it's a dynamic process, and something changes that balance, whether it's chemical, another pathogen interferes, but then you've lost the balance, and now it is no longer the immune system alone or the pathogen alone, but it's the combination of the two, and somehow you have to reset the homeostasis or that balance. So let's first talk about the cohort, because this is the topic today, is to look at difficult cases. Now, we heard a lot about this cohort last night and today, so I won't belabor it other than to say that in the cohort from the outbreak of Incline Village in Dan Peterson's practice, a significant subset of those, 77 of those to date, have presented with an immunological defect, if you will, known as a clonal T-cell receptor gamma rearrangement. And that means it's an innate immune response. It's not the typical rearrangement of the alpha-beta T-cell. And that's suggestive of a persistent viral infection that you can't clear, or as Dan said earlier, even the development of lymphoma. These patients also have the concomitant low NK cell function, so another area of the innate immune response is dysfunctional. They also have high RNA cell activity or dysregulated RNA cell. RNA cell is an antiviral endonuclease that is involved in recognizing those RNA that Dr. Chia talked about and degrading them and causing the infected cells to die. And those innate immune defects are hallmarks of this particular group of these of patients of the most difficult to treat. So again, these studies were isolated RNA, DNA, plasma, and peripheral blood cells from 100. We call these our top 100. So in the viral array, this viral microarray profiling was led by Dr. Frank Rossetti, longtime colleague at the Cancer Information Program at the National Cancer Institute. His colleague in that endeavor is Dr. Rachel Bagney, and she's with the Advanced Viral Technology Program, and she actually developed the custom microarray that I'll describe, and us. So the problem is, prior to this study, no study's been able to address the expression of all the viruses in a single sample from a CFS population with immunological phenotypes suggesting a chronic acute infection that I just described to you. So we addressed this using this custom oligonucleotide array that Rachel developed, and it can detect all known mammalian and avian pathogenic viruses. Each virus is represented by 10 or more features of 72 base pair oligonucleotides in conserved regions, in variable regions, early and late. There's redundancy that allows us to identify new viruses that may arise via recombination. With regard to human influenza and the avian flu viruses, it's a tiling technique they use such that you can identify even viral subtypes as well as the precise definition of influenza recombination events. They're highly sensitive. You can detect RNA and DNA particles at the level of 5 to 10 or 20. I did these all with RNA, so we're looking at expression. So some key points in talking about these large arrays, these gene expression arrays, and particularly the viral microarray, it's really important to understand the data analysis. These are very complex data sets, and some confounding issues and assumptions have to be considered. That is that the detected virus is not necessarily the causative virus for the tumor or disease, but it could be a consequence of impaired immunity, which we've alluded to today. Only a portion of the tumor or disease population actually get the intended virus. So that confounds things as well. As Dan said, 25% of his population have HHV6A. Importantly, the control group has to be very clean. You simply, you have to have not contact controls and not somebody with a cold. You really need a very clean control who's not sick, because you're looking at the same viruses, particularly herpes viruses. They've co-evolved with us for millions of years. So we all have these herpes viruses, or most of us have these herpes viruses. So when you're looking at differential expression patterns, you have to consider that. We use multiple probes in the arrays, and they're all designed to detect the same virus. So you're not looking at just one thing, you're looking at different points of the virus so you can see the total replication, if you will. And we've used a two-class contrast, disease versus control, and a sample level enrichment-based pathology ranking method that was developed by my friend Bob Stevens at the National Cancer Institute Biocomputing Center, and I'll describe that in a few minutes. But the conventional gene level array analysis method really not suitable for these kinds of tasks because of the variabilities just between individuals in general and the cycling of these viruses even in patients. So the rationale for the analysis then was to look for the virus detected by multiple probes with good signals, but with the biggest contrast, the largest difference between the two classes, healthy and disease. So, and again, clean control samples are essential. So this is the method, the schematic workflow of how it works. What you do is you generate your two classes, your controls and your disease. And you look at the data distribution across all of the gene sets, about all of the samples, and you can see the controls versus the disease. There's a big difference between the two classes when considered this way. You also get sample level differentiated genes for each single sample, so you could look at each single patient as an individual. And then you batch compute these, which I wouldn't dare to do, and merge the batch result into the matrix and for the enrichment scores. And then what you can do is you can actually evaluate the actual ranking against the distribution in the ranking scores and get meaningful data as to really what's going on between the difference of the disease and the control. And the reference there for Bob Stevens is published last year. He actually developed this method just to look at these arrays, because when they analyzed them the first two times, it was just crazy. Spike samples weren't there. At any rate, so this is one that I wanted to talk a little bit about EBV, HHV4, and this is looking at all 64 probes. So there are 64 probes on that for that gene on this array, or for that virus on this array, and you can show the hierarchical clustering analysis and just individual ranking that Jonathan showed us earlier, where the green shows a low level of virus and the red meaning it's hot. And the arrow indicates, sorry, they still think I'm at the National Cancer Institute, the tumor, or the diseased person, is the red arrow indicates areas where there's high level and big differences in expression in a single person. So these individual patients have then, EBV is one of their issues. So you can look at these data as a class and try to make conclusions about the class of disease versus healthy, but you can also get a lot of valuable information out of an individual. So I'll show you again a little bit more of the sample ranks, and this shows you some of those confounding issues. If you look at this sample, 2 CFS patients versus a control, when you simply rank them in the most expressed, as Dan said earlier, the single biggest thing when you look at these data just from an eye is the patients express tremendous amounts of virus. The same viruses are expressed by the controls and even in the same rank, but the level of expression is considerably lower than in the patients in these particular experiments. So again, in this case, if you just do individual ranking, the top 7 viruses were herpes virus CMV, HHV4, EBV, and then some endogenous retroviruses and other herpes viruses predominate throughout this group of patients. So again, interestingly, when you look, if you're just ranking the viruses, the top 6 viruses in all 5 CFS patients, when you individually rank, was a human endogenous retroviral DNA. So the human endogenous retrovirus was what was the key if you just look at individual ranking. If you take it then and do the class contrast data analysis that I showed you, ranking the method lower for 4.1, if you consider that, in the first experiment we did, it ranked very highly, as I just said. But because in the control samples it also ranked highly, it was in the top 9 in the controls as well, which immediately reduced its significance to the disease causation using that class contrast method and put it at rank not in the top 9, but actually in the top 1,500, showing that as far as the disease is concerned, or suggesting as far as the disease is concerned, that wasn't the issue there. So finally, to conclude this particular study, the top 22 virus by that pathway ranking method are shown here, and in that cohort, CMV, HHV5, and HHV7 were the top 2 ranking viruses in the ME/CFS with the clonal T-cell receptor gamma rearrangements, or gamma delta T-cell clones. Interestingly, again, need to know your technology. Dr. Bagney came to me, and I was looking for HHV6A, and I said, "Well, Rachel, why isn't there?" And she said, "Well, the sequences that were chosen for that particular virus on the array were in multiple repeat areas, and they were not." They were more highly variable instead of the constant regions. And she said, they wouldn't design it that way again, because if you actually go confirm that, you can see that CMV was followed by HHV6, HHV7, and EBV, where they were all identified as the most highly expressed herpes virus in the CFS cohort. So these data suggest that multiple highly active virus infections can distinguish CFS patients with certain immunological innate immune deficiencies in the pathogenesis. So how do we translate that? As we heard, I believe it was Malcolm say, well, we can reduce these chips now to develop custom arrays. So we've built a custom array that is essentially 4, 5, 6, A and B, and 7. And it's a single chip, and you can look at all of these at exactly the same time. And so this chip will be used will be licensed by the company VIPDX for clinical use, but essentially any company can license this chip, and it's validated with controls on the plate. It's a 1-plate assay, 1 amplification. So this herpes chip and the cytokine signature diagnostic tools, which I'll talk about in the next study, can be used worldwide, and then if we all isolate the samples the exact same way, we can translate that to other cohorts and use these technologies. So that's the translational end of the experiment. So next I'm going to talk about the cytokine and chemokine profiling, the immune component of this study, and that study was led by Dr. Vincent Lombardi of the WPI with help from the University of Nevada. Dr. Relman, Wei Yang, and Julie Smith-Gagan are marvelous statisticians who did the data analysis. The patient samples were provided by Dr. Diamond and Peterson, and we, of course, always thank the patients and controls who eagerly volunteer to participate. So the rationale for this study comes from the last study we did, and that is that chronic innate immune activation by pathogenic triggers in a genetically susceptible host, and we'll show you that in a minute, can mediate the pathogenesis through a cytokine-chemokine storm. So what we did here was we used multiplex suspension arrays. So that's antibodies on beads in a single well where you need 50 microliters of plasma or serum, and you can look at 30, it's like an ELISA in suspension. You can look at 30 different proteins in the sera all at the same time, all in the same sample, which gives you the opportunity really to analyze the complex relationships of these proteins as they're in the body expressed out of different cell types from both the innate and adaptive immune response, who of course talk to each other regularly. So I wanted to elaborate a little bit more. Dan showed you this slide earlier, but the rationale for the cytokine study becomes more clear when you consider what this clonal rearrangement means. We've said it's persistent, it's suggestive of persistent active infection, particularly CMV. And it's also predictive of the development of lymphoma. And that's because CMV actually encodes a gene that actually turns these cells on, encodes a protein that actually turns these cells on. It's antigenic for them. So these gamma delta T cells, then, that's what you get if you rearrange the gamma chain first. They're known to play an active role in the resolution of pathogen-induced immune responses. So they accumulate at sites of inflammation. So as Dan also said, sometimes sampling in the periphery is not a good thing because we can't see these T cells when we look for them. But we can see the evidence in the cytokine and chemokine patterns that are left behind because when they're turned on, they're expressing the chemokines MIP1 alpha beta and the cytokines TNF alpha, IL-10, and gamma interferon. So again, the clinical criteria for testing that Dan used was the acute viral onset of CFS and the lymphadenopathy and splenomegaly along with the gamma delta T cell rearrangement. So here are the results of the study. We had 118 patients and 138 controls, and clearly you can see a number of cytokines and chemokines. These might be all chemokines. A number of chemokines all upregulated, and you can see that it makes sense with their role. IL-8 is actually upregulated not only by CMV infection. It exists in a loop where CMV turns up IL-8 and IL-8 turns up the expression of CMV. So exactly what we saw on our microarray is what we see here. MIP1 alpha and beta are key neurodegenerative, they're targeting molecules that actually go to the brain and have receptors on neurons by themselves. Again, all of these p-values, they're highly significant in a fairly large sample size. So, and all of these chemokines, IP10 is an interferon response protein, again, suggests an inability to handle an infection. So if we look then at the functional T cell subsets, thank you, John, for that nice description, so I don't have to talk about them. But what we've heard about before is that in CFS, there is a Th2 shift. Well, clearly this subgrouping and looking at the sickest of the sick or the different groups show you that there is not a Th2 shift. In this population, Th2 responses are highly significantly down-modulated, where the Th1 response and the Th17 response, that's IL-1 beta and IL-6, are key regulators of a Th17 differentiation response. And again, these cells are found at its sites of inflammation, so you won't see IL-17 in the blood, and we did not. So other cytokines that are dysregulated that fit, of course, interferon alpha was downregulated. And it's important in that it stimulates both the macrophages and the NK cells, which we know are defective. So this might be one reason why they're defective, that they are not making enough alpha interferon to elicit that antiviral response. IL-1RA is a key regulatory molecule for IL-1 beta. It really has to dampen that response fast, as you saw how high our IL-1 beta is, and unfortunately it's downregulated here. IL-7 stimulates some of the unusual B and T cell subsets that I won't have time to talk about today. But again, when you look at these things in concert together, they make sense with the pathology of the viruses we just saw. A hierarchical clustering shows you the power of this technique for subgrouping. And you don't need to know anything about what's up here, but you can see a different pattern here with the clonal rearrangements over the CFS patients without clonal rearrangements. Lots of red up in this region of these cytokines and chemokines I just described. Control number 14 is a little sick, so we'll have to find what's going on there. But again, low levels of these cytokines and chemokines in healthy individuals. So you can cluster these and determine subgroups and see subgroups right away. So the random forest analysis, another strength of being able to use these powerful statistical analysis, and I won't go into this one, it just uses a tree-like unprone classification where it doesn't bias the data at all, and it says who correlates with who in what algorithm, what is running together or correlating together to in this response that you're seeing across the whole thing. And it's really uniquely suited for the cytokine and chemokine analysis because it can handle the highly skewed data that we see and the variation among individuals quite easily, and it can weight the contribution of each cytokine and chemokine according to its relatedness with others. And so I'll show you with that technology, that statistical technology, we could identify a 5 cytokine chemokine signature that could be used diagnostically if validated that would predict a CFS patient 94% of the time, only 6% false positives, and similarly only 6% the sensitivity, only 6% false negatives. Easily done, translate it to any lab, and you can look at those cytokines and chemokines in your patients. Interestingly, in this part of this study, we did a cancer fatigue group as a control, if you will, a disease match control, and they did not have this signature. They have a different signature. We need numbers to work it out at this point. We need more of those patients. So finally, I'm going to talk about the genetic profiling. And this study was done in the Carrington lab at the NCI, again, Cancer Information Program, under the direction of Mary Carrington and Pat Martin. And all the work I'm going to talk about was done by a talented research associate in the lab, Sarah Bass. So the human leukocyte antigen is the most highly polymorphic or disease-associated area of the genome. More diseases are associated with polymorphism in HLA than any other gene region. So the polymorphic immune response of these genes affects the level of the host resistance against pathogens. So when we've recently discovered the killer immunoglobulin regulatory receptor on these cells as well, and it's a binding ligand for HLA, and I'll talk about that because together, combined with other molecular genetics and related immune response genes, these changes lead to disease. So HLA class 1, quick lesson. When a host encounters a foreign particle, it enters the cell, antigen-presenting cell. It's chewed up into little peptides, and it's presented along with class 1, its binding partner, to give to the T-cell receptor, where a very specific T-cell receptor recognizes that HLA in the context of that peptide, and then it generates the cytotoxic T cells, the adaptive immune response, such that you get an adaptive killing immune response to that antigen very specifically. So we looked at all the Sarah looked at the associations for HLA, B, and C, which are the major regions known that are variable, in 97 controls and 67 cases. And she found no significant association at the 95 confidence level for HLA-A. But if you look at some of these with the higher odds ratio, 2.9, 1.12, 1.5, with higher numbers of those in those particular allotypes, we might see something significant, but because of the number of allotypes in the group, we didn't see any significant associations. We've added 200 samples, 100 from, or 200 from Jonathan's set and cohort, and another 150 from ours, so we should get some interesting and see if those actually fall out to be significant. There was also no significant association for HLA-C, and again, this particular odds ratio of 2.56 with bigger numbers could hold true to indeed be a susceptibility allotype because the odds ratio is higher that you'll get the disease if the cases is higher than the number of controls, whereas down here you might have a protective effect of that allele. Interestingly, we found HLA-B44 was significantly associated with increased susceptibility to ME/CFS at a 2.56 odds ratio. And I know there was an HLA-B, I don't remember exactly which one, associated with parvovirus infection, and I know Jonathan published that several years ago. So again, there may be an HLA-B44 allele associated with increased susceptibility to ME/CFS. So there are more than 50 allotypes of the B44, so they did a fine genetic mapping out to 4 sites and didn't see any difference between the subtype specificity with the B44 association. Finally, we're going to look at NK cells. We've heard a lot about natural killer cells, and they're another lymphocyte that reacts with HLA, binds to HLA in order to elicit a functional response. So normally if the target cell, every cell in the body every nucleated cell in the body expresses HLA class 1. So if you're a nucleated cell and you're happy and you're just doing fine, then you will meet your binding ligand, the KIR receptor, and you won't lyse. It'll inhibit a lysis of the target cell, saying that's a healthy cell. But in tumor cells and virus-infected cells, they often downregulate their class 1 or lose their class 1, such that this binding and inhibitory reaction doesn't occur, and you get lysis of the target cell because your immune system says that's a bad guy, get rid of it. So we wanted to look at the KIR associations, the HLA class 1 ligand binding groups for these KIRs. And there are 2 in HLA-C1 that are dimorphisms of the 80 asparagine 4 lysine. And what we see here is that NK cells, it's a balance between activation and inhibition. You can activate them, and they'll kill, or you can inhibit that killing and they won't. And you see here with HLA-C1 subgroup that it highly favors, with the blue area, the KIR 2DL2 and 2DL3, saying that it's going to inhibit that killing. That NK cell is not going to kill. Similarly, the lysine will bind to a binding partner of KIR 2DL1, and that is again inhibitory. The HLA-B4 has also a similar dimorphism with the threonine and the isoleucine, and you see that both of those KIR binding ligands favor that inhibitory KIR 3DL1. So, suggesting we know there's NK cell defects, we know these NK cells don't kill. It's not always a level of low perforin, so it could be a genetic susceptibility to have more of the inhibitory KIRs expressed. So we looked at those, and we found indeed that the HLA group 1, CW group 1, when it's homozygous, is highly protective of CFS. If you have that homozygosity, you won't get CFS at a significant p-value. Whereas the heterozygous situation of C1 pairing with C2 is actually associated with increased disease susceptibility. Finally, if we look at the BW4 alleles, they're both associated with, BW4 is associated with increased disease susceptibility, suggesting, again, that on a genetically susceptible host, you won't get the appropriate immune response to fight the pathogen because they're inhibited. So the summary is that HLA-B44 is significantly associated with susceptibility to ME/CFS. There was no significant association with HLA-A or C, though there are trends which suggest there could be. HLA-BW4, a ligand for KIR, is associated with susceptibility, as is homozygosity at the CW1 subgroup, associated with protection, while heterozygosity is associated with increased susceptibility. And as I mentioned, we're doing lots more samples right now in a different cohort to see if this same type of approach holds up. So in conclusion then, I think taken together, these data suggest that a systems biology approach to a translational research project is an effective strategy for the development of serum biomarkers that can stratify subgroups of ME/CFS patients for appropriate anti-inflammatory, immune-modulating, antimicrobial, and antiviral therapeutics. So you can use these to follow therapy. You can use those to suggest the appropriate therapy. And then finally, Annette mentioned earlier that my students work long hours and they get punchy, so they develop these acronyms because my long career at the government, we do a lot of acronyms. So as John showed us, the CDC, they came up with can't, don't care, and based on our bias in the studies I've just shown you, they want to rename CFS to it's the virus stupid. Thank you for your attention.